Welcome back to this video lesson on Meselson and Stahl's experiment, which is an experimental proof for the semi-conservative DNA replication. And the learning goals are: 1. Explain the semi-conservative model of DNA replication with simple illustration. 2. Determine why N15 and N14 isotopes were used by Meselson and Stahl in their experiment. 3. Represent the elegant experimental design of Meselson and Stahl. And 4. Interpret how the experimental results of Meselson and Stahl provided evidence for semi-conservative DNA replication. While proposing the double helix structure of DNA, Watson and Crick realized that the complementary nature of base pairing in the DNA strongly suggested for a semi-conservative model of DNA replication. They proposed that when the two strands of the DNA get separated, these strands would act as templates for the synthesis of new complementary strands. Here's a simplified illustration of an uncoiled double-stranded DNA, which are both shown in black color. So when the two strands of the parental DNA get separated, each of the parental strand would act as template strands for the synthesis of new complementary strands. And the two resulting double helices, or the two resulting daughter DNAs, would each be containing one old strand and one new strand. So after replication, every new double-stranded DNA would be a hybrid consisting of one old strand and one new strand. One old strand and one new strand. And such a mode of DNA replication is known as semi-conservative DNA replication. And this was the initial proposal of Watson and Crick in the year 1953. Semi-conservative replication was not the only model of DNA replication proposed in the mid-1950s. Two other prominent models were also put forth, which was the conservative model of DNA replication and dispersive model of DNA replication. It was the elegant and timeless experiment conducted by Meselson and Stahl in the year 1958 that provided the experimental proof for semi-conservative replication to be the correct model of DNA replication. Meselson and Stahl studied DNA replication in E. coli bacterium by using two isotopes of nitrogen. So they used the rare and heavier N15 isotope as well as the common and lighter N14 isotope to distinguish the old and new strands in the DNA. They also used cesium chloride density gradient centrifugation technique so as to separate the DNA bands based on their density. I'll briefly explain some of the facts which was already known to Meselson and Stahl before they actually went forward with their experiment. They knew that normal E. coli DNA has N14 isotope, which means the natural nitrogen isotope in E. coli is N14, and this is applicable to all of the living cells. They also were aware of the generation time of E. coli, or the time that is required for the E. coli to reproduce. E. coli doubles every 20 minutes. In every 20 minutes, they would be completing one replication cycle. And through their pre-experimental work, Meselson and Stahl came to realize that DNA can be separated in cesium chloride density gradient medium. So when DNA is added into cesium chloride density gradient medium and then centrifuged, DNA gets separated by forming a band or bands at the position where the density of the DNA matches with the density of the cesium in the solution. Now suppose if both the strands of the DNA has N14 nitrogen isotope, then such DNA would be separating, or such DNA would be forming a top band in the cesium chloride gradients. This is because N14 is a lighter isotope. Whereas if both the strands of the DNA are having N15 nitrogen isotope, then such DNA would be separating, or such DNA would be forming a bottom band in the cesium chloride gradients because N15 is the heavier isotope. So before we proceed into the details of their experiment, I would encourage you to remember the position of the N14 DNA band and the N15 DNA band in the cesium chloride density gradients medium. Let me now walk you through the elegant experimental design of Meselson and Stahl. they started off by culturing E. coli bacteria in a medium that had heavier N15 isotope in the form of N15 labeled ammonium chloride medium. And this N15 labeled ammonium chloride medium would act as a source of nitrogen for the newly synthesized DNA. As a matter of fact, Meselson and Stahl grew E. coli in N15 medium for up to 14 generations to allow enough time for the creation of E. coli population that had all their DNA labeled with the heavier N15 isotope. And in this way, they were able to first create the parental generation or generation 0 by culturing the E. coli in N15 medium for 14 generations. They then isolated the DNA and mixed the DNA sample with cesium chloride solution to measure the density of the DNA through centrifugation. And as expected, in their result, they obtained a single band of DNA which was located at the bottom position of the cesium chloride solution. This showed that after 14 generations in the N15 medium, the DNA composition of generation 0 or parental E. coli were all pure N15 DNA, which is to say that both of the strands in these DNA had N15 heavier isotope. And here in my illustration, I have used red color to indicate N15 isotope. So what you see here is both of the strands of the generation 0 DNA have red coloration, indicating that both of the strands are containing the heavier N15 isotope. So it is a pure N15 DNA. Next, they transferred such N15 labeled E. coli cells into a medium that had the lighter N14 isotope in the form of N14 labeled ammonium chloride medium. And from now on, N14 medium would be acting as the source of nitrogen for the newly synthesized DNA. And you need to note that Meselson and Stahl isolated the DNA sample from the N14 medium after every 20 minutes because they knew that in every 20 minutes, E. coli cells would be completing one replication cycle. So after 20 minutes, the first generation DNA sample was isolated from the N14 medium and was mixed with cesium chloride solution to measure the density of the DNA. And in their result, what they obtained was, they again obtained a single band of DNA. But unlike the position of the G0 DNA band, generation 1 DNA band was located in the halfway position of N15 density and N14 density. So from this result, Meselson and Stahl inferred that after one cycle of replication, all the DNA molecules are now a hybrid DNA, which is to say that each DNA was composed of one N15 strand and one N14 strand. And because it was a hybrid DNA molecule, so the density of the DNA is now an intermediate density between the density of the heavier N15 and the lighter N14. When they isolated DNA samples from N14 medium after 40 minutes and centrifuged in cesium chloride solution, in their result, they obtained two bands of DNA of equal thickness. One band of DNA appeared at the intermediate density position, just as in the case of generation 1, and the other band appeared at the top N14 density position. So from this result, Meselson and Stahl inferred that after 2 rounds of replication, 50% or half of their daughter DNAs were hybrid DNA and the other half were pure N14 DNA. And this result from generation 1 through 2 was enough for Meselson and Stahl to confirm that DNA replication is semi-conservative in E. coli. To understand how we have equal amounts of hybrid DNA and pure N14 DNA produced after 2 rounds of replication, let's trace back the parents of generation 2, which are the generation 1 hybrid DNAs. each hybrid DNA has one N15 strand and one N14 strand. So during the second round of replication, these two strands in the hybrid DNA get separated, and each strand would be acting as a template for the synthesis of new DNA strand. Now the daughter cells that received N15 templates are going to be synthesizing new DNA strands in the N14 medium, thereby producing two molecules of hybrid DNAs. While the daughter cells that received N14 template would be synthesizing new DNA strands in the N14 medium, and this would be resulting in the formation of two pure N14 DNA molecules. So after 2 rounds of replication, we have a total of 4 DNA molecules produced, of which 2 of the DNA molecules are hybrid DNA and 2 of the DNA molecules are of the pure N14 DNA types. So this pattern of replication follows with or conforms with the semi-conservative model of DNA replication. I have illustrated and explained Meselson and Stahl's experiment up to generation 2 in N14 medium, since this is enough to confirm the semi-conservative nature of DNA replication in E. coli. Nonetheless, I would encourage you to continue this experiment for 2 more generations, as Meselson and Stahl actually did, to figure out the proportion of N14 or light DNA and the proportion of hybrid DNA after 60 minutes and 80 minutes respectively. And just in case, here is an overview of Meselson and Stahl's centrifugation result up to 4 generations. To sum up, semi-conservative DNA replication refers to a mode of replication in which the DNA strands separate and each act as a template to make a complementary copy of itself, so that each daughter DNA molecule has one old and one new strand. Following the Meselson and Stahl's experimental evidence, many scientists confirm semi-conservative DNA replication in different species as well. Semi-conservative DNA replication is indeed a universal mode of replication in all living organisms. I'll come up with my next video on the key enzymatic mechanism of DNA replication.